Hello everyone. Today I am going to explain recombinant DNA technology, which is a form of genetic engineering used to produce useful human proteins, such as insulin. Recombinant DNA is DNA that has been created by joining together genetic material from two different species. At GCSE level, the main example of this technology is the production of human insulin using genetically modified bacteria. Insulin is a hormone that regulates blood glucose concentration. People with type 1 diabetes do not produce enough insulin because their immune system destroys the insulin-producing cells in the pancreas. As a result, they require regular insulin injections. The process begins by isolating the human insulin gene from a chromosome. This gene contains the base sequence that codes for insulin. The gene is removed using enzymes called restriction enzymes, which cut DNA at specific sequence. A plasmid, which is a small circular piece of DNA found in bacteria, is also removed from a bacterial cell. The insulin gene is then inserted into the plasmid. This plasmid now contains DNA from two different organisms, making it recombinant DNA. The recombinant plasmid is then inserted back into a bacterium. This bacterium is now genetically modified and can express the human insulin gene. As the bacteria reproduce by binary fission, they copy the plasmid and produce insulin protein. The genetically modified bacteria are grown in large containers called fermenters, where conditions such as temperature, oxygen levels, pH, and nutrient supply are carefully controlled to maximize insulin production. The insulin produced is then extracted and purified to ensure it is safe for human use. This method allows insulin to be produced in large quantities, at a low cost and with a high level of purity. There are several advantages of recombinant DNA technology. The insulin produced is chemically identical to human insulin, so it reduces the risk of allergic reactions. It is also more ethical than using animals, and provides a reliable supply for patients worldwide. However, recombinant DNA technology raises ethical and environmental concerns. Some people worry about the release of genetically modified organisms into the environment or the long-term effects of genetic engineering. For this reason, strict regulations and safety procedures are used. In conclusion, recombinant DNA technology involves cutting, joining, and transferring genes to produce useful proteins such as insulin. It plays a vital role in modern medicine and demonstrates how genetic engineering can improve human health. That's the end of today's video. Thanks for watching.